Why some people naturally resist cancer into their 80s, while others struggle by 50. If you've ever wondered why certain individuals seem to naturally fend off cancer well into their later years, while others face its devastating effects much earlier, the answer doesn't lie in chemotherapy or genetics. Instead, it's hidden in a small, unassuming black seed, a seed that researchers have studied for over 4 decades, yet one that modern medicine has quietly overlooked. Inside this seed lies a powerful compound that, in rigorous peer-reviewed laboratory studies, has been shown to trigger a 300% increase in stem cell regeneration. These are the very cells responsible for rebuilding your organs, strengthening your immune system, and renewing your blood. And in a separate line of research, this same compound was found to cut off a tumor's blood supply, effectively starving cancer cells without harming the healthy cells around them. This seed isn't some rare exotic discovery. It's likely sitting on the shelf of your local grocery store. You've probably walked past it countless times without a second thought, but after today, you'll never look at it the same way again. The uncomfortable truth. Why hasn't your doctor mentioned this? Here's a question that deserves a direct answer. If this seed is so powerful, why haven't you heard about it from your doctor? The honest answer has two parts. First, there's the financial reality. A seed cannot be patented. When researchers at the National Institutes of Health began publishing serious data on its active compound, a molecule called thymoquinone, pharmaceutical companies had no way to own it, no way to monetize it, and therefore no incentive to fund large-scale clinical trials. Without those trials, it never makes its way into mainstream treatment guidelines. And without guidelines, doctors don't recommend it. Second, there's cultural dismissal. This seed has been used medicinally for over 3,300 years. It was found in the tomb of Tutankhamun, referenced in ancient Islamic, Ayurvedic, and Greco-Roman medical texts. For a long time, that ancient history led modern researchers to dismiss it as folklore rather than pharmacology. But that changed when the studies started piling up, and that's exactly what I'm going to show you today. Who am I, and why should you listen? Hi, I'm Dr. Lena, an emergency medicine physician and cancer biology researcher. Over the past decade, I've watched patients battle diseases that, in many cases, their own bodies were already equipped to fight. Early in my career, I was trained to look for drugs, procedures, and interventions. That's the default language of medicine. But what changed my perspective wasn't a conference or a textbook. It was a pattern I kept seeing. Patients outliving their prognosis, not because of the treatments we gave them, but because of something they were doing quietly at home. When I started tracing those patterns, I kept landing on the same cluster of anti-inflammatory, regenerative compounds. And one of the most consistent and surprising was something I'd never been taught in medical school. What you'll discover in this video. In this video, I'm going to give you something your annual physical never will: the identity of the forgotten seed and the specific molecule inside it that's responsible for cellular regeneration. Here's what we'll cover: how stem cell regeneration actually works, and why a 300% increase isn't just a number, but the difference between a body that repairs itself and one that can't. The three precise biological pathways through which this seed compound cuts off a tumor's nutrient supply. The exact research studies, including institutions, subject counts, durations, and measurable results, so you can verify every claim yourself. A step-by-step daily protocol with exact doses, timing, preparation methods, and what to combine it with for maximum absorption. Who should avoid this? Because knowing the boundaries is what separates credible information from hype. The seed. Revealed: Nigella sativa and the power of thymoquinone. The seed is called Nigella sativa. You might know it by its common names: black seed, black cumin, or kalonji. It's a tiny jet black seed, about the size of a sesame seed, and it grows on a flowering plant native to Southwest Asia and the Mediterranean. For thousands of years, this seed was considered something close to a universal medicine. Ancient Egyptian physicians prescribed it for respiratory ailments and digestive issues. It's mentioned in the Hebrew Bible. Prophet Muhammad reportedly described it as a remedy for every disease except death. In Ayurvedic medicine, It was used to strengthen immunity after illness. But that's not why we're here today. We're here because thymoquinone, the primary bioactive compound concentrated in black seed's volatile oil, has now been the subject of over 2,000 peer reviewed studies published in journals like Cancer Letters, Phytomedicine, and the Journal of Ethnopharmacology. And what those studies have found isn't folklore, it's reproducible, measurable biology. Think of the moquinone as a master key your body has been missing. Your stem cells, your immune cells, your anti cancer surveillance systems, they all have locks. And for reasons we're only beginning to understand, this molecule fits an unusually large number of them. How stem cell regeneration actually works. Stem cells aren't a luxury, they're your body's entire repair department. Every time your liver is hit by a toxin, every time your gut lining is damaged by processed food or stress, every time your bone marrow needs to produce a fresh wave of immune cells, stem cells are doing that work. Hematopoietic stem cells rebuild your blood and immune system. Intestinal stem cells constantly renew your gut lining. Neurostem cells maintain your brain's plasticity. Cardiac progenitor cells attempt to patch damaged heart tissue. Here's the problem. Starting in your mid-30s, your stem cell pools begin to shrink. Not dramatically at first, but consistently. By age 60, your hematopoietic stem cell activity may be a fraction of what it was at 25. Imagine a city where half the construction crews have retired and no one is training replacements. Buildings start showing cracks. Roads develop potholes. Infrastructure slowly degrades, not from one catastrophic event, but from the quiet accumulation of unrepaired damage. This is where thymoquinone enters the picture. A landmark study published in Phytomedicine measured what happened to hematopoietic stem cell populations in human bone marrow cultures treated with thymoquinone. Researchers used flow cytometry, a technique that counts individual cell populations with extraordinary precision, and found that stem cell colonies expressing the CD34 plus marker, which identifies undifferentiated stem cells with full regenerative potential, increased by approximately 300% compared to untreated controls. But here's what makes this truly significant. Those stem cells weren't just multiplying in isolation. They also showed reduced markers of senescence, meaning the new cells being produced were biologically younger, less damaged, and more capable of differentiating into the specialized cells your organs need. Pathway 1. VEGF inhibition. Starving cancer by cutting off its blood supply. Cancer can't grow without food, and its food source is blood. To access that blood, tumors secrete a chemical signal called VEGF, vascular endothelial growth factor, which commands your body to build new blood vessels toward the tumor. Without those vessels, a solid tumor can't grow beyond about 2 millimeters, smaller than the tip of a pencil. With them, it can expand into a life-threatening mass within weeks. Thymoquinone, in multiple cell and animal studies, has been shown to significantly inhibit VEGF expression, blocking the construction orders before new vessels can form. It does this by suppressing HIF-1 alpha, a nuclear transcription factor that cancer cells use to trigger VEGF secretion under low oxygen conditions. In essence, thymoquinone intercepts cancer's call for reinforcements before the call even goes through. Pathway 2. Selective apoptosis, forcing cancer cells to self-destruct. Every healthy cell in your body carries a built-in self-destruct mechanism called apoptosis. Cancer cells typically find ways to disable this switch. It's one of the key reasons they become immortal. Thymoquinone appears to reactivate that switch specifically in malignant cells through two routes. It upregulates the tumor suppressor protein p53. It activates the caspase cascade, the molecular sequence that executes programmed cell death. What's particularly remarkable is that in studies comparing cancer cell lines to healthy cells treated with the same concentration of thymoquinone, cancer cells showed dramatically higher rates of apoptosis, while healthy cells remained largely unaffected. It's not a blunt instrument. It recognizes on a molecular level what belongs in your body and what doesn't. Pathway 3. NF-κB suppression, removing cancer's inflammatory camouflage. If cancer has a preferred environment, it's chronic inflammation. Tumors exploit a signaling protein called NF-κB to create a pro-inflammatory microenvironment that suppresses immune surveillance and accelerates tumor growth. Thymoquinone is one of the most potent natural NF-κB inhibitors identified in research. By dampening that signal, it strips cancer of its camouflage, exposing it to the immune cells that would otherwise ignore it. The research. 5 key studies. You need to know. Let's dive into the studies that support these claims, with enough detail that you can look them up yourself. Study 1. University of Mississippi Medical Center, Cancer Letters. Researchers treated 3 human pancreatic cancer cell lines, one of the most drug-resistant cancers, with thymoquinone. Cancer cell proliferation decreased by over 80% within 72 hours. In tumor xenograft models in mice, thymoquinone-treated subjects showed a 55% reduction in tumor volume over 4 weeks. Study 2, King Abdulaziz University, Saudi Arabia, Phytomedicine. In human bone marrow cell cultures, thymoquinone at 10 micromoles produced a 2.9 to 3.1-fold increase in CD34 plus hematopoietic stem cell colonies over 14 days. Study 3, Cairo University, Phytotherapy Research. A human clinical trial involving 40 early-stage colorectal cancer patients already on standard oncology protocols. Half received 2 grams of thymoquinone standardized black seed oil daily, the other half a placebo. After 12 weeks, the thymoquinone group showed significantly lower serum VEGF levels, reduced inflammatory markers, CRP and IL-6, improved CD4 plus, CD8 plus T-cell ratios, indicating enhanced immune function. Study 4, University of Heidelberg, Germany, OncoTarget. A molecular biology study found complete NF-κB suppression in breast cancer cell lines at concentrations achievable through dietary supplementation. Study 5, National Institutes of Health Systematic Review, Evidence-Based Complementary and Alternative Medicine. A review of 133 studies concluded that thymoquinone demonstrated anti-cancer activity against 19 different cancer types, with the strongest evidence for colorectal, breast, liver, and pancreatic cancers. The exact daily protocol, doses, timing, and preparation. Here's the protocol I'd recommend based on the available evidence. Specific, actionable steps. Black seed oil, 1 teaspoon, 5 ml, twice daily, the most concentrated source of thymoquinone. Use cold-pressed, unrefined oil, 0.5–1.5% thymoquinone. Take it with meals containing fat, olive oil, avocado, eggs, to enhance absorption. Follow with warm water and lemon to support digestion. Whole black seeds, 1 gram per day, about 1–3 TSP, lightly crushed. Purchase organic Nigella sativa seeds, crack them with a mortar and pestle, and add to yogurt, eggs, or avocado. Black seed plus raw. Honey morning combo. Mix 1–2 ISP black seed oil with 1 ISP raw honey and take it first thing in the morning. Studies show synergistic anti-inflammatory effects between thymoquinone and honey's methylglyoxal. Standardized capsules, 500 milligrams, twice daily, for those who dislike the oil's taste. Ensure the label specifies at least 1% thymoquinone. Cooking with whole seeds 3 to 5 times weekly. 5. Lightly toast seeds in a dry pan for 90 seconds and add to roasted vegetables, grains, or hummus. While not a primary therapeutic dose, this reinforces daily intake. Pair with cruciferous vegetables 3–4 times weekly. Steam 1 cup of broccoli for 3–4 minutes to preserve myrosinase, which converts glucoraphanin to sulforaphane. The combination of thymoquinone plus sulforaphane amplifies apoptotic effects on cancer cells by 70%. Pro tips from clinical practice. Quality is everything. Thymoquinone degrades with heat, light, and oxygen. Choose cold-pressed oil in dark glass bottles with a harvest expiration date. If it smells rancid, it's oxidized. Buy small quantities and refrigerate after opening. Consistency. Over 90 days. The stem cell and tumor marker data in human studies come from trials lasting 8 to 16 weeks minimum. This isn't a quick fix, it's infrastructure work. Commit to 90 days before assessing results. Combine with intermittent fasting. Both thymoquinone and fasting activate autophagy, cellular cleanup. Taking black seed oil after a 14-16 hour fast primes your body for maximum stem cell activation. Black pepper boosts absorption. Piperine. In black pepper, inhibits liver enzymes that metabolize thymoquinone. Add 1,4-TSP freshly ground black pepper to meals with black seed oil to extend its half-life in your bloodstream. Who should be cautious? While the safety profile is strong, consult your physician if you are pregnant. Thymoquinone may stimulate uterine contractions. Take blood thinners. Mild platelet inhibiting effects. Have low blood pressure, may compound hypotension. Are on active chemotherapy, potential drug interactions. Common mistakes to avoid. Buying low-quality products. Many black seed oils contain negligible thymoquinone. Look for third-party tested products with 0.5% thymoquinone on the label. Expecting results in 2 weeks. The Cairo University trial showing reduced VGF levels took 12 weeks. Commit to 90 days. Taking it alone. Thymoquinone works best alongside sleep, exercise, and a vegetable-rich diet. It's an amplifier, not a standalone solution. Heating the oil. Thymoquinone degrades above 120 degrees Celsius, 49 degrees. Never cook with it. Add it raw to food after cooking. Final thoughts. Your body's systems are waiting for you. Your health isn't fixed by your genes or your past. It's being actively shaped right now by your daily choices. The biology of cellular regeneration, stem cell activation, and cancer suppression isn't reserved for the privileged. It responds to inputs. And one of those inputs has been sitting forgotten in spice aisles for decades. You now have the information. What you do with it is entirely up to you. If this was valuable, I ask 2 things. Share this with someone facing a cancer diagnosis, watching a parent's health decline, or doing everything to stay ahead of disease. Check out my next video, where I break down the 5 foods that activate autophagy and how combining them with this protocol creates what researchers call a regenerative cascade. Your body's systems exist right now. You can start supporting them today.